يعطيكم العافية طلابنا الأعزاء. Uh, we're starting with our sixth immunology lecture. Um, so some concepts to review and move forward from. We recall that T cells on their receptor, they can only recognize peptides, and this is for the default or for the majority of conditions. T cells only recognize antigens that are polypeptides, and only when those are presented on MHC proteins. So MHC proteins has to be the presenting factor here. So the restriction of CD4 cells and CD8 cells to bind MHC1 or MHC2 is due to the fact the binding site on the T cell receptor is actually what predilects them to either becoming an MHC2, an APC, and only finding CD4, or إذا كانت CD8, it can only bind to MHC1. So that's what makes them. It's the binding sites on the T cell receptor that forces them to only interact with one or another. With the appropriate MHC proteins, okay. So CD4 can only bind MHC2, and then the CD4 will stabilize the connection, and the same thing for CD8. تمام. Furthermore, CD8 and 4 which stabilize the connection further aid in this restriction اللي هو the CD4 can only bind to MHC2 and so on. So MHC1 are usually used to present intracellularly produced antigens. Why? Because I want cytotoxic cells, cellular immunity, to be the presenting factor here, be the dominant factor. So obviously MHC1 are going to present only intracellularly produced antigens. حكينا إنه MHC1 usually only shows what is inside the cell. هل هو normal proteins normal، يعني بتاخذ عينة من هالبروتينز داخل الخلية وتطلعهم. إذا كانوا مثلا abnormal proteins due to a mutation or cancer, the cell should be killed. Viral proteins مثلا, and so on. هلا MHC2 proteins usually present antigens of extracellular origin, right? So APC that have or APCs that have taken up مثلا bacteria, taken up fungi, whatever, and broken down. Obviously, these things were from outside the cell, so these are extracellular sources. So this is a further distinction that we make between MHC1 and MHC2. MHC2 will present extracellular antigens, whereas MHC1 will present intracellular antigens. MHC1 is based in the cellular immunity, whereas MHC2 is with the regulatory part of the adaptive immunity. Tamam. So this is why when I give a vaccine composed of dead viral cells, it will not cause a CD8 response. Because why? Because this will not be presented on MHC1. So an antibody response as these viruses will not be presented on MHC1. So they did not replicate and infect cells and then presented on MHC1. So this will be presented on MHC2. And obviously I will then have a B cell humoral response. So that's why the majority of vaccines will become extracellular antigens, which are taken up by APCs, which are then presented to CD4 cells, and then produce a Th2 or a humoral response. So you will have already made antibodies that will trap the virus before it enters the cell, and that's the majority of your defense that you get from the vaccine, which is actually, you will see that it is actually protective. So next time you meet the virus, you have antibodies. It will intercept and bind these viruses and then disabling them before they can even enter the cells. So this restriction is due to the distinction in the acquisition and processing of these antigen. هي نقطة نتوسع فيها اليوم أكتر إنه between intracellular parts and extracellular parts. So the restriction between MHC1 and MHC2, intracellular versus extracellular, is due to actually the distinction in their acquisition, and then very نقطة مهمة كتير in how we process these proteins. So an endogenous protein يعني viral protein, cancer protein زي ما حكينا، البروتين انت عم تعمله بجوا الخلية. So it will be processed in different compartments داخل الخلية that will ultimately within the cytoplasm that will ultimately restrict them and mark them as these are intracellular proteins within the cytoplasm, and then they are different in compartments than those that are derived from extracellular sources. فها مثلا the association of endogenous foreign proteins داخلية بروتينات داخلية مع MHC1 happens ليش؟ due to the difference in processing. طيب where these proteins are cleaved by a proteasome. هذا موجودكم صورة. So when you cleave these proteins and you destroy them, you break them up into smaller parts, and then هدول البيبتيدز are chaperoned by what is called a TAP transporter. This TAP transporter transports this protein من خلال الrough endoplasmic reticulum, where it is linked there in the rough endoplasmic reticulum with MHC1. And that you will see that this is completely distinct from the processing of extracellular proteins. So the endogenous protein MHC1 complex هلا now travels to the Golgi. بكمل زي كانت بتصنع بروتين داخلي نفس الطريقة، يعني بتدخل من الرف للجولجي وبتكمل قاعد. So and then to the cell surface where it is presented. هلا in short, MHC1 presented proteins go through the typical endogenous protein manufacturing process. So as like we said, as if you are making a protein. So انت قاعد بتقطع البروتين الداخلي، بتكسره، بتدخله، بتمشح البروتينات، بتحطها مع ال MHC1، بتكمل لانه you are also producing MHC1 as well. So and then you link them together, and that source and that process is completely distinct and completely separated. مهم النقطة هاي completely separated from هلا نحكي عن البروسيس الثانية if proteins that are derived from extracellular sources. هلا نحكي عن proteins that come from an extracellular source. هل هدول proteins come from extracellular sources? Obviously more likely these will be taken up by phagocytes. الخلية العادية ما راح تاخذ will not take up extracellular proteins. This is the job of a phagocyte. So إذا كانت neutrophil ما عندها it does not have MHC2, but البقية will have MHC2. So هل هدول extracellular proteins مش أنا بدي أخذهم، أنا بدي أكسرهم. My job, I'm not trying to present them with my normal proteins. What I'm trying to do is I got these الخلايا, bacteria, whatever virus, whatever I got, I want to destroy it. So I'm not going to send it to my Golgi, to my rough ER. I'm going to put it into an endosome. وبتعرف تذكروا phagosome, and then I want to put in some enzymes and destroy these organisms. So we will see that the proteins in an endosome, and this is where the peptide fragments will be linked with an MHC2 protein into a complex, and then from the endosome the peptide MHC2 complex migrates to the cell surface, and then that will be presented. Okay, so from the endosome حكينا the peptide MHC2 complex will migrate to the cell surface, and that's يعني the main idea here. So let's discuss a question here. Why don't the intracellular proteins, which some of those are going to get degraded, why don't we ever present them on an MHC2 complex? So there is a protection mechanism راح نورجيكم صورتها هلا that is placed that mostly prevents نشوف راح في small communication that mostly prevents endogenously produced proteins from being linked with an MHC2 protein. هاي أول نقطة. النقطة الثانية, as you are breaking down a protein, how is the most unique part of the protein selected? كيف بدك تعرف كيف الخلية وهي قاعد تكسر بهالبروتين with just with different enzymes. يعني the enzymes, they don't care. They're going to break down bonds and then break down the protein to the smallest part that they can. كيف بيعرفوا? How do they know which is the unique part? Actually, they don't know. What happens إنه the protein is not just a sequence of amino acids. It is actually folded in the secondary and tertiary structures, right? And every protein is unique. كل بروتين شكله مختلف ولا otherwise all proteins will have the same function. So if all proteins are unique, when I break down this protein that has these many secondary and tertiary structures which are unique again, when I break it down, I will find that my enzymes find it hard to reach the areas of the protein that are heavily folded secondary or tertiary structures. فلما أقطع البروتين, these remaining parts that my enzymes could not get into will actually be the most unique part of the protein, and that is what is usually presented to the MHC molecules. تمام. So خلينا نكمل. So this occurs due to the presence of an invariant chain attached to MHC2. هذه protection mechanism that proteins when they are not inside the endosome. So if they are not inside the endosome, هو الاندوسوم هو المكان أنا بكسر فياته exogenous protein. So when MHC2 proteins are being produced somewhere else, أكيد I will produce them with my endogenous protein, because MHC2 in the end of the day it's an endogenous protein. protein أنا بصنع فياته. لما أنا أصنعه, when I make the MHC2 protein, they put a lock عليه, بسكر عليه, بحط عليه قفل. When the MHC2 protein travels to the endosome, the enzymes in the endosome will actually break the lock, and only then that the MHC2 will be able to bind to extracellular proteins in the area اللي هي endosome where I am actually breaking down these extracellular proteins, and that is the main protection mechanism of how I prevent extracellular proteins أو عفوا intracellular proteins from binding to my MHC2 complex. شو بهمني بهالنقطة كلها؟ Because if I am presenting بدور على CD4 cells وبحط على ال MHC2 indigenous protein, زي كأني I'm poking a bear, زي كأني بروح على الدن or شو اسمه يعني بيت دب وقعد شو اسمه بوخذ فيه بعصاية بصحي فيه. So why am I trying to find a CD4 cell that can react to my cell proteins, even though I trained them? I might find one that by mistake might have not met this protein or something, and then I will have an autoimmune disease. So that's why I put this protection mechanism to make sure only extracellularly derived proteins are ever bound to MHC2 protein. So no proteins will be able to attach to MHC outside the endosome. The lock mechanism is only removed for the MHC2 protein within the endosome only with these enzymes inside the endosome. تمام. فهاي الصورة اللي حكيناها, كل اللي حكيناه so far is what we just mentioned. So هاي الاندوسوم, خلينا نحكي عن extracellular بالبداية. So وآخر شيء نقرأ هاي اللي هنا نحكي عنها. So phagocytosis, I am bringing an extracellularly derived protein. أخذتها البكتيريا, كسرتها. So I broke it down. So now it is now a late endosome. هيها كسرتها داخل الاندوسوم. In the late endosome, this is where the MHC2 molecules are present. بكسر هدول. I find a unique part. I have all these enzymes. I find a unique part. I bind it to it, and then it is sent to the cell surface. A lysosome, the same thing. You're taking up these different proteins. You break them down, and then they merge here as well, and then you can also present these extracellularly derived proteins as well. So that is the main mechanism. So هلا هنحكي عن intracellular proteins. Intracellular protein حكينا with the main mechanism of production of عادة normal proteins are produced here. They go to the plasma reticulum, they are folded, they go to the Golgi. This is where MHC1 is present. So as you have intracellularly derived proteins موجودين جوا الخلية, you break them down with a proteasome. فعندك self proteins. You might have some exogenous proteins that exit the phagosome, and this is where the crossing between the two pathways can occur. مش endogenous على MHC2, exogenous على MHC1. That's different, which is good actually, because I might want to see if this cell is infected by something. This is a protection mechanism to see if I am infected by something that is not being picked up. So في عندي crossing between the two pathways here, هي هون. but it is not of endogenous proteins on MHC2, it is on exogenous protein on MHC1. So obviously if I do that معناته هاي الخلية has something wrong, or يعني it can be tolerated depending on the protein. So تمام. Viral protein هو extracellularly derived بس goes with the intracellular mechanisms. Self proteins exogenous protein كلهم بتكسروا بالبروتيسوم زي pepper grinder كلهم بيطلعوا and they are broken, they are sent to the ER, they are packaged with the MHC1 and then they are sent to the cell surface, and that's how we this is the two compartments of the mechanism and the mechanism of how you present extracellular and endogenous proteins. There's one point where you can have an autophagosome, which might show some of your self proteins here. But you see this is a mechanism ممكن يعني this is something that you can look up of cathepsins that may prevent these proteins from being presented to MHC2. So I don't want to get into this too much يعني at our level بكفينا اللي حكيناه so far. So خلينا على everything else apart from this mechanism, from this communication mechanism. So MSC1 can bind peptides derived from exogenous proteins internalized by endocytosis. هي وجهناهم هنا. This is the phenomenon called cross-presentation, where specific subsets of dendritic cells are particularly adept at this هاي اللي هنا. Mediating this process, which is critically important to initiation of a primary response by naive CD8 cells. Obviously, that makes sense. And this is actually beneficial, which would allow cytotoxic response against certain cells that have taken up certain toxic material, toxins مثلا. So that's the story here. طيب, the B cells. So how is there a different story? They don't have a T cell receptor. So B cells, the story is a bit different. These cells interact with their surface immunoglobulin, IgM, صح؟ ما عندهم T cell receptor. So they interact with foreign molecules with their B cell receptor, with their IgM and IgD. And obviously حكينا IgM and IgD can bind to non-peptides. So how does this communication happen? So since the antigen presentation with MHC2 is not needed to activate B cells, حكينا not always, you have a T cell independent pathways, so ذكروها, and the ability of B cells to present antigens which are not always peptides, which ultimately activate themselves indirectly by presenting the antigen to a CD4 cell. ذكروا حكينا هاي قصة الهابتن. So in contrast to MHC2 antigens, in contrast to MHC2 antigen presentation, where it can only present to peptides, هلا حكينا. So IgM and IgD antigen receptor can recognize non-peptides as well. And as we saw before, this is how haptens can bypass the requirements of how small they are and how they are not proteins by conjugating them to carrier proteins. And then to be associated with MHC2 antigen presentation, the carrier proteins peptides are instead used in this case bypassing the third requirement, ذكروها, which is the peptide presenting to CD4 and MHC2, and then this will activate the CD4 cells, and then I will have a T cell dependent response, T cell dependent B cell activation at this point, and then I can even class switch my antibodies and then make IgG or something else. So the signal transduction, antigen MHC complex on the APCs, they interact with the T cell receptor of whichever T cell receptor is specific to this protein. The effort of this interaction is to produce a signal sent from the T cell receptor, ذكروا حكينا CD3 protein, from the T cell receptor to the nucleus. I want to get a nuclear response out of this. What signal happens? The signal is transmitted حكينا ذكروا the CD3 protein, which detects if the T cell receptor is occupied, and then it transmits the signal down to the nucleus. هلا حكينا نحكي المحاضرة القادمة نحكي how it happens. This is where it happens. So CD3 is a part of the T cell receptor. تمام. حكينا it's bound with every T cell receptor on the خاصة على the CD4 cell and complex through the pathway eventually to the influx of calcium. That's the first signal. So once the T cell receptor is occupied, an influx of calcium will happen. This influx causes a specific protein activation اسمه protein calcineurin. سهل نحفظه من calcineurin. فكروا نيورين يعني نيورو اللي هو زي تحكي عقل الخلية. So the nucleus. So calcium بده يودي السيجنال to the nucleus. So calcineurin, this is a serine protease or often serine phosphatase, and it exerts its enzymatic function in the nucleus. The main point حكينا what is the integral here is I want to switch on the genes for interleukin 2. تذكروا حكينا if you activate interleukin 2 then you have genetic activation of interleukin 2 and then you are always forever reacting to this antigen. So switch on the genes for interleukin 2 and interleukin 2 receptor, and now we know that this calcineurin, now we know how this calcineurin how it functions, so we can remove MHC2 mediated immunity by blocking calcineurin. So if you make a drug اسمه cyclosporine تعرفوه. So this is a very well known anti-inflammatory drug and can be used for many diseases, autoimmune diseases, and even organ transplantation. And then this will hinder the cell immunity in many cases. So why does block only cell mediated and not antibody mediated? Because you have blocked interleukin 2, which you will need for the blocking, for the maintenance of Th1 response, right? And interleukin 4 and 5 will be for the antibody mediated, which may be done by only a T cell independent response, if you recall. So interleukin 2 is produced by CD4 cells, which is activated by antigen presenting cells presenting on MHC2, and interleukin 4 from unknown sources is what activates the Th2 response. That's the answer to our question. So let's look here. This is fairly low yield. يعني إذا بدك ما في يعني ما راح تطلع من كثير منه أسئلة مثلا امتحانات بورد أو امتحانات whatever. What we need to know, we will try to mark that the points where you might be asked about anywhere else. So first النقطة اللي بدنا نعرفها إنه the ultimate thing is بالنهاية I want to activate the genes on the interleukin 2 locus. This is very important to know. واحد. اثنين, I have two signals that I need together. تذكروا I need signal 1 and signal 2. The co-stimulatory signal and the binding of the T cell receptor to its antigen, and then the CD3 اللي هو حكيناها will send CD3 will send the signal down, will initiate the signal down to the nucleus. هاي مع هاي will produce the ultimate signal. هلا إذا بتجمعهم اثنين مع بعض you will see بالبداية phospholipase C1 which induces the calcium influx. Calcium influx will bind to calcineurin and calmodulin. هاي النقطة هون الثانية مهمة. And then you will have a lot of other proteins that can also be activated. Both calcium influx, calmodulin, calcineurin مع the other enzymes خاصة phosphokinase C will initiate the nuclear factor of activated T cells called an NFAT, and then هي هنا واحد مع اثنين will activate this, which will other enzymes مع AP1 مثلا will turn on the gene for the interleukin 2 locus, which will ultimately start its transcription, and then you will have production of interleukin 2 from that cell. So now that cell is activated against this specific antigen. تمام. So let's see if we have missed anything here. Stimulatory signal, co-stimulatory signal, phospholipase C, which induces calcium influx, in fat, the pathway, and then the protein kinase with the nuclear activator protein AP1. هدول هم اثنين مع بعض اللي هو حكيناهم. And then both together will activate interleukin 2 production. So you see, if you do not have the co-stimulatory signal, so if you have signal 1 only without signal 2, this will result in anergy. So you don't have any response because you're not going to induce the protein kinase C. So that will break down the chain, and then you will not have the recruitment of NFAT and AP1 and so on. Come on. So it is in this step, interleukin 2 production, that clonal proliferation will happen. So as we mentioned, and then this makes you always have a memory against this antigen and always producing interleukin 2 against it. So because you have activated your genes against this antigen. So meaning if the CD4 cell reaches the interleukin 2 production, it will always proliferate and make clones of itself. So interleukin 2 is always known as T cell growth factor. Stimulates the helper T cell to multiply into a clone of antigen specific. So also stimulates CD8 cells. زي ما بنعرف, the majority of these cells will carry out their effector and regulatory function. And then from these clones, a few we will see that they are kept away as memory cells, and then everything here we know. Activated CD4 positive T cells also produce another lymphokine called gamma interferon. We know this, which enhances the ability of APCs, خاصة macrophages, making them produce more MHC2 proteins and thus present more antigen. This enhances the macrobiocidal activity of macrophages. It is important to know that the activation of T cells is not all or none. There is a gray area of partial activation. Tamam. So we know full activation and full complement of lymphokines that are released. And then what is partial activation? So partial activation leads to a release of fewer lymphokines. That would lead to a weaker response and less cells and less clonal proliferation, less everything. So this is dependent on the epitope. So that was used in the activation. هو الابيتوب اللي انت حطيته على التي سيل ريسبتور عشان تشغل السي دي 3. So it depends on this epitope actually of how strong a response do you make. So how does this happen? The explanation for this is, and this explains why some people can clear certain infections more than others. It's just your genetics and your T cell response, your T cell receptor that you have made against different things. So as our cells have 3 genes for class 1, تعرفوا, and then we have many genes for the class 2 locus from each parent. So you have 6 possible copies for each gene in class 1 and 6 copies making many copies for class 2 proteins. So MHC2 is now able to present peptides with different amino acid sequences. This is something we touched on before. Memory T cells, tamam. With these cells, we are able to mount a quicker, effective response to antigens even years after. Why years? Because these are cells. I will continuously replenish them. يعني إذا كبرت بالعمر الخلية, it will divide and make new, fresh 2 cells, and that's how they keep maintaining themselves. This is not, proteins can't do that, obviously. Protein, إذا بقدم, it will be broken down. That's why your antigens, your antibodies, will only survive for a few months at best. So this memory response to a specific antigen is due to several features. In the first exposure to an antigen, there are few cells responding to the stimulus. So they start from 0, نعرف هذا, and then you have to go all the way to the CD4 cell activation. So on the second exposure, you will have many cells that are already produced, and then you will activate them and you replicate them. And then you will also make more memory. So each time you meet them, your memory gets stronger and stronger and stronger. So that means this whatever antigen in the environment is a common antigen. It's something that always wants to attack me. So I want to have a larger response, always waiting for it. بعكس مثلا antigen, I met it only one time in my life. So why should I have a high memory for it? And that's how it depends on how many times you meet it. The more you meet it, the more memory you make, and the more and the stronger the response each time it will be. So cells, as opposed to antibodies, they can live for many years. We know this. What else? A memory cell has a special ability which allows it to have much stronger whole exaggerated response to smaller amount of antigen, and would require less co-stimulation. This heightened response is because these cells produce higher amount of interleukins than regular naive cells. هذا شو بيحكي لك؟ بيحكي لك إنه how do memory cells take us from memory back to full response? It's because their ability to produce interleukins and signals is much heightened. It's much stronger than a normal T cell. This way it will drive the force and speed of reaction towards the invader much quicker and much stronger than إنه خلية T cell عادية. Because it says I want to go back from مثلا أنا الميموري T cell أنا مثلا إذا ببلش لقيت antigen جديد I want to start from 0 all the way to 50, which is meeting the T cell CD4 cell, and then from 50 to 100 I am making my response, making clonal proliferation, antibodies, cells, whatever. So خلصنا الانفكشن, I go back to 50. This is where my memory is. هلا I want to كمان هذا من 50 to 100 I want to even that I want to reduce it. This is where the memory T cell does it exaggerates the response once it is activated to bring us to a quicker response back to 100 again. Now we will discuss the T cell receptor and how it actually does its job that we are discussing from the previous slides, and the next slide we will talk about. بذكر حكينا إنه how do I make a variety of T cell receptors, each respond to a different antigen. حكينا the way that we do this is by having different pools of genes, and these different pools باخذ منهم كل واحدة قطعة and then I come up with a different piece زي كأنه ليجو. عندك مثلا خلينا نحكي مثلا 50 قطعة حمراء كل واحدة شكل، و40 قطعة صفراء كل واحدة شكل، ومثلا 30 قطعة خضراء كل واحدة شكل. تأخذ واحدة حمراء واحدة صفراء واحدة خضراء تركبهم مع بعض تطلع بشكل مختلف. And that's what happens with the T cell receptor. So and then they are composed. The T cell receptor itself is even composed كمان إضافة of 2 polypeptide chains. Each chain obviously will have to be produced differently, which are then associated with the CD3 protein, as we know. So in order for the T cell receptor to detect many different antigens, هذا اللي حكيناه, we take these pools. So the genes that code for the T cell receptor chains are made by the rearrangement of multiple regions of DNA, which creates variability. شو هدول البولز الليجوز الخضراء والحمراء والصفراء؟ The gene loci هم أسمائهم the variable هدول الأولى. الثانية اسمها diversity genes. الثالثة اسمها joining genes J, and then الأخيرة constant. It's actually constant عند الكل. Why do I want a constant thing? Why don't I make it all variable? Because I have a constant reaction بالنهاية with the T cell receptor, صح؟ والantibody the same thing. Every antibody will at the end wants to be bound to يسموه FC receptor that is bound by اللي بتعرفوا عليه الفاجوسايت مثلا. So I want all phagocytes to يتعرفوا على FC receptor. كل FC receptor راح يكون constant. The same thing for the T cell receptor. I want a constant part for me زي ما تحكي هذا part that my body wants to respond to and reacts to. So it's constant. بقية ال T cell receptor is not constant. It's variable. هو بعمله من V and D and J parts. ال J بربط ال variability and diversity genes مع ال constant genes. So all of which work together to provide high variability and diversity, which you can even have almost 100 million different receptor proteins that can be made of T cell receptors. The variable regions have hypervariable domains and even more diversity at the end of the protein. And the two genes هدول الراج 1 والراج 2 بيطلعوا ترى بالامتحانات، انتبهوا لهم. RAG1 and RAG2 they encode recombinase enzymes that catalyze this gene rearrangement, are similar in T cells and B cells. فهي شكله تذكروا شفناه. This is the alpha chain and this is the beta chain, and each this is the diversity and variability genes joining عادة بده يكون هون, and then you have the constant part which you are going to put here. So مع CD3 مربوط مع CD4. So let's take a closer look. We have actually 70 or more, between 70 to 80 variability genes هم الأحمر, and then I have diversity genes. Actually it's the other way around. The joining genes are the more dominant and the diversity are less. So you have about 61 joining genes, and then you have fewer diversity genes, and then you have the constant gene واحد أو اثنين بس. So the T cell receptor alpha locus موجود بchromosome 14 consists of these 70 to 80 variability segments, each preceded by an exon. Obviously this is what I'm coding for the leader sequence, and then a cluster of 61 J gene segments, and then you have diversity genes, and then you have a single C gene for the constant A region, which contains separate exons, the constant region hinge domain, and a single exon coding for the transmembrane and cytoplasmic regions. اللي هو حكيناه هنا. So these transmembrane اللي هو دائما بحطوا كل الخلايا هذا ما بدي أغيره. It's not diverse. I want it to always be the same. So T cell receptor beta locus مختلفة, موجودة على chromosome 7. It has different organization, 52 V genes, and then other numbers of 6 or 7 J genes and a single C gene. So this will provide even more diversity, that I have different genes for each locus alpha chain or beta chain. So let's look. مثلا طلع هيك صورة ثانية اللي هون الألوان بتعبر عن which part of the gene is brought. So variability هنا مثلا البرتقالي, الألفا جين مثلا هون موجود لون ثاني. So this is for the beta gene, مثلا الأخضر الغامق الأحمر الأزرق. All of this together will make all this part of the T cell receptor, which is the beta chain, and then the same parts will be made in a similar manner for the alpha, and then the MHC will come and bring this this antigen to this receptor. Another picture showing how they are rearranged من alpha وبيتا ألوان مختلفة ثانية, C, G, J, D, and V, the same thing. So from previous figures we can deduce that each T cell has a unique T cell receptor on its surface. تمام. With hundreds of millions of different T cells, we have different, each will have a different specificity. Each unique T cell once activated, معناتها هاي variability مهمة لي. It detected something in my environment, I need it. So I will do clonal proliferation, مهمة, to yield a large number of cells that are able to deal with this antigen. Antibodies have a similar genetic rearrangement, راح نحكي عنها شيء ثاني, but it's the same concept. So T cell receptor, even though it is uniquely specific to an antigen like an antibody, it differs from antibody in two ways. It has 2 chains rather than 4. شوفوا الantibody has 2 alpha chains and 2 beta chains, or 2 heavy chains and 2 light chains. So it recognizes antigen only in conjunction with MHC proteins. The antibody, it can react to anything, not just peptides and MHC proteins. So this is where B cell or T cell independent activation is appreciated, because B cells don't use T cell receptor, but they use immunoglobulins, antibodies, as their cell surface. So they can bypass the need for MHC as well. Also, the T cell receptors are always anchored to the outer membrane, whereas I can secrete the antibodies. ما فيش circulating form of T cell receptor. So monomeric IgM is the B cell membrane, and the pentameric والخمس قطع مع بعض, this is the actual one that circulates in the plasma. So it's the very large antibody. This is why it doesn't cross the placenta, it can't go to the placenta. That's why in the placenta داخل البيبي only have IgG. effects of super antigens on T cells. حكينا عن المحاضرات اللي هلا خلصناهم الاسبوع الحالي اللي هو we talked about TSST, whether it's staph or strep, these super antigens, these toxins that cause toxic shock. So in contrast to a typical antigen, which activates only a few helper cells, a super antigen, it's super. Why? Because it activates a large number of helper T cells all at the same time. It has the effect of causing a very high, profound activation of many different helper T cells. ما بعرف شو إذا لما نحكي many different helper T cells, it بتصير unique لإلهم كلهم. So everyone will react to this antigen, and then everyone will think, oh, I have found a unique T cell to this one. You will have a very high amount of T cells, helper T cells activated, and then you will release a very high amount of interleukins and cytokines, which will produce a shock response, which is مسميه toxic shock. So this toxic shock syndrome, toxin binds directly to MHC2 proteins without the internal processing of the هلا بورجيكم صورتها. So a super الانتيجين العادي شو بصير له؟ الانتيجين العادي اخذته, you processed it, you found a unique part, بتروح على MHC2 بتحطه بتركبه, هي T cell receptor with the V and J and D and whatever, found it, و CD4 و CD3 كل هالحكي. This is the normal way. A super antigen goes and binds from the outside of the T cell receptor. It feels like to the helper T cell that this T cell receptor is occupied. It is not occupied. وكمان it binds to the MHC class 2 in the same manner. فهذه بتروح بدها present it, وهذه بتحكي I am occupied. But this actually is not unique to either. لا هو unique لل MHC2 ولا هو unique لل T cell receptor. And then you will have, this will happen to many different helper T cells, and you will have obviously ما صار في processing. So this is quick. هذا مش quick. هذا this is slow. I need processing, we'll find a unique part and present it, we'll find. This will go directly to the immune system, bind different APCs, and then the APCs will all go and find all helper T cells are binding to this, and they will have an exaggerated, very high response to this toxin. So that's why we mentioned here. It binds directly to the class 2 MHC protein without internalization. This is very fast, and this complex interacts with variable portions of the beta chain, of many of the variable beta chain مهمة بيجي امتحانات, meaning these antigens have regions that are detectable by many different T cell receptors, and this will give you a heightened, exaggerated response, very high amount of interleukin 2 and other interleukins. Some other, حتى مش بس بالبكتيريا, some viral proteins can also have super antigen activity. كتير toxins موجودة تعمل هاي الطريقة. تمام. Now I think we finished the majority of what we want to cover, the new parts that we want to cover in this lecture. Now we want to cover some يعني accessory materials here and there. So let's هو إنه يعني ما حكينا إنه just to cover our tracks about T cells, the features of T cells and B cells والخلايا اللي حكينا بنحكيهم so far. So we know that T cells make up the large number of circulating lymphocytes, تقريبا من 65 ل 80%. البقية كلهم B cells. Within lymph nodes, T cells are found in the inner and subcortical regions. تمام. T cells have a very long half-life. Mitogens, so these cells can be stimulated to proliferate by mitogen, a mitosis generating molecule, mitogens. So mitogens for T cells, مثلا في hemagglutinin, concavalin A, endotoxin مثلا. All these are mitogens that can produce proliferation of T cells. So T cells are detected in blood by using sheep blood. Sheep blood. The T cells will all bind to this RBC, تجيب RBC and يسموها rosette. So these are lymphocytes. بدك تلاقي lymphocyte وتحط RBC من sheep, and you find all the lymphocytes will bind to it and to a rosette manner. This will tell you these are all lymphocytes. So effector functions شو نعرف إحنا so far. This is for you to look at. So we know TH1, TH2, TH17, and then we have the CD8 cell. TH1 hypersensitivity against intracellular. TH2 protects against worms. تعمل لك antibody response. TH17 in the gut, and CD8 for the cellular response. TH1 cells, macrophages, we know, are the main effectors of cell-mediated immunity. This is very important for intracellular organisms, TB, histoplasma, مثلا. What else we have that we mentioned in bacteriology so far. حكينا عن الكلاميديا والريكيتسيا مثلا. So the main signaling molecule in this time is gamma interferon. Again, this is activating, helps to recruit macrophages, and the actual function of destroying these intracellular pathogens is performed by the macrophage itself. So it's well studied that the case of MTB حكيناها is a specific ذكر حكينا interleukin 12 which indicates that the foreign protein was found, and interleukin 12 is an inducer of the correct type of this response in this case, which means I want to make a TH1 response and rather than a TH2 response, which would be useless in this manner. So this interleukin 12 gamma interferon axis is very important for our ability to activate a TH1 response and have a correct activation and then destruction of these intracellular organisms. فمثلا هدول الصور من histoplasmosis, intracellular coccidiosis, TB طلعوا acid fast bacilli inside a cell. TH2 حكينا these cells مع eosinophils هم effectors of reactions against helminthes كثير مهمة, worms. So eosinophilia and IgE is the main way of how you destroy these worms. The most important interleukin نعرفه هو 4 and increase the production of IgE. IgE binds to the surface of the worm, and then eosinophils that bind to the heavy chain of IgE, and then they keep secreting enzymes. As they are bound, they keep destroying these worms. TH17, we know it attracts neutrophils to the site of infection, whereupon the neutrophils beach, and then they can destroy the bacteria that are invading. CD8 شو أشياء يعني نراجعها هون. CD8 cells mediate the cytotoxic response, virus infected tumor cells, abnormal cells, graft rejection. So I think everything here we mentioned so far, there's nothing new. How do they kill? We know perforance. They form channels through the membrane, which obviously these holes will leak out the cell's contents and kill it. Granzymes and proteases. So these are degradative enzymes, and they can work against the proteins in the cell membrane and lead again to loss of cell content. Granzymes كمان بيعمل caspases. They activate the caspases, which is the same mechanism inside the cell that will initiate apoptosis. بتعرفوه هذا from your molecular biology course. So cytotoxic T cells have no effect on free virus. This is important, only on the cell that is producing the viral protein. So what is the main apoptotic mechanism which T cell kill their target? Is that they activate apoptosis through the Fas ligand interaction. تعرفوا هذا from your pathology course. And this apoptosis mechanism happens when the Fas, which is a protein displayed on the surface of many cells, and the cytotoxic T cell recognize this epitope. The Fas ligand is then induced in the cytotoxic T cell, and then this will result in the apoptosis of this cell. NK cells also employ the same mechanism as well. So to eliminate virus-infected cells, we require an antibody-mediated mechanism that can result in the destruction of cells. So this is a new concept موجودة بهاي السلايد. So now we have a connection, a bridge between humoral immunity and cellular immunity اللي هو I not only flag up extracellular materials to be phagocytosed اللي هو مثلا IgM or IgG and then I باجي أنا كphagocytic cell وباخدها. I can direct cytotoxic T cells to go and destroy cells that are bound with the antibody. So يعني إذا بدك تفكر فيها it's really just improve phagocytosis بطريقة يعني that you which is still the same as the cellular immunity, but you are enhancing it with the antibody on the cell surface. Basically it's يعني the same concept that we mentioned before, but هلا نحكي why we are delineating it by itself. So in an antibody dependent cellular cytotoxicity, this is a combination between IgG and phagocytic cells. An antibody directed at the virus is bound to the surface of the infected cell. تمام؟ هلا أنا شو ال delineation عندنا هون؟ أنا NK cell بروح على أي خلية ما عدا MC1. تمام؟ أنا CD8 cell بروح على خلية that is exhibiting on MHC1 صح؟ exhibiting on MHC1 their viral proteins بقتلها. هذا أنا صرت إيش؟ cytotoxic cell. cellular immunity. هلا الإضافة اللي هون بهاي السلايد the antibody dependent cellular toxicity is where the phagocytic cell will go to that. هلا أنا بعمل IgG، الIgG ممكن يرتبط بالانتيجين الموجود على MHC1 يعني it found its antigen, right? I'm still producing the antibody against the virus يعني الموجود خارج الخلية. So when this antibody binds to the cell, this cell is flagged to be destroyed by macrophages. Here macrophages will come and then will kill the infected cell. So the bound antibody is then recognized by phagocytic cells, macrophages, NK cells, by an IgG receptor on their surface, and the infected cell is killed. So هاي هي من أهم mechanisms of the antibody-dependent cellular cytotoxicity. If I cover a worm with IgE, كل هدول السيلز اللي حكيناها قبل شوية, all these cells will come and bind the IgE with their constant receptor and start releasing مثلا هون كانت eosinophil. So not phagocytes are the affected, the eosinophil will just keep pumping out its enzymes and destroying this worm. So this is antibody-mediated or antibody-dependent cellular cytotoxicity. So the actual killing is mediated by the granules and not by... So you can see it's similar to CD8 cell killing, just the effector cell is different. هذه هي النقطة المهمة الأولى. الثانية it's not MHC1 dependent, it is dependent by the antibody. And this way I can direct هاي النقطة مش مكتوبة هنا. I can direct cellular cytotoxicity against شو? Against non-polypeptides. Because what am I targeting now? I'm using an antibody which can target non-polypeptides. So this can direct my phagocytes, my eosinophils, my NK cells or whatever towards cells that are infected by foreigners that are not exhibiting peptides. مثلا هذه كمان نقطة إضافية بنضيفها هون. هلا مثلا in tumors عندك new antigens، انت بتعمل antigen جديد it's not self and it's displayed by the MHC1 surface. CD8 cells will recognize this and then stimulated by the interleukin 2 will target these tumor cells and destroy them. So interleukin 2 production means that this clone of CD8 cells will proliferate, which will mean this clonal expansion is now able to kill this type of tumor. So this phenomenon is called immune surveillance. So obviously if this happens, هل أنت immune to this type of cancer؟ الجواب نعم ولا. نعم بطريقة إنه you are immune to this stage of the cancer. هلا if the cancer progresses further in other cells, then you will need to produce a new reaction. واحد. اثنين, cancer cells might progress further, become further deformed, and then they are not even displaying anything, or they are, تذكروا حكينا بالمحاضرة الماضية, they are causing energy. So that's another way of doing it. But in the early stages, this clonal expansion will kill the tumor cells. هلا إذا تستخدم أنت allograft, CD8 cells will recognize this class اللي هو MHC1 on the surface of the foreign cells. And then حكينا هون this is where we do HLA typing, and they might reject or accept this MHC1. So who activates the CD8 cells in an allograft rejection? They need interleukin-2 production by CD4 cells, right? So they will recognize the foreign class MHC2 molecules on certain cells in the graft, APCs from the graft, macrophages, lymphocytes, and then that might actually present them with the antigen they need to be activated. Then they secrete interleukin-2 زي ما حكينا, stimulates CD8 cells, clonal expansion, and then you are forever immune against this graft. So you will destroy it. There's no خلاص ما فيش حل هنا. الحل الوحيد هاي الكدني بدك تكبها هاي الأقصى شو خلاص gone. The skin or whatever you are using as an allograft. So this means now the body has memory against this allograft, and there is very little that we can do at this point. So okay, let me stop here.